While I'm sharing my slides, I want to say, first of all, thanks to the Siegel Rare Neuroimmune Association for organizing this. I want to thank the Kennedy Krieger Institute and Hopkins Medicine for hosting. And also I want to thank Carlos Pardo, who's been a real driver behind the AFM group and these meetings especially. So thank you for inviting me and thank you for all your work. So I'm going to talk a little bit about the virus and the cells, and my lab really focuses on what goes on inside a cell when a virus infects, and we focus on Picornaviruses. And in recent years, my lab has really turned to looking at EVD68 as our primary interest. So what I would define my talk as is trying to explain why cellular membranes might be interesting to people who are interested in clinical outcomes when we're talking about a non-envelope virus. And usually when you talk about non-envelope viruses, we're not particularly interested in membranes. But we've in fact found that it has a pretty important role in pathogenesis and host dissemination, in our opinion, and I'm going to hopefully convince you of that over the next few minutes. So I just want to remind you that Picornaviruses have remarkably conserved genome organization. So much of what we've learned about one Picornavirus, like, for example, Coxsackie virus, we can apply to other viruses because their genomes are very similar. So I will spend most of my talk talking about EVD68. I will occasionally talk about rhinovirus or poliovirus. I want to assure you that everything we've looked at so far, enterovirus D68 has been a very typical Picornavirus inside the cell, so that we think that we can apply a lot of the lessons we've learned elsewhere while extending them and really now using EVD68 as our primary virus model. EVD68 also physically looks like the other Picornaviruses. These are some space-filling models of poliovirus. You can see EVD68 in the upper right. They're very simple capsids. They're only made of 3 or 4 proteins in a repeating structure. And they are, as I said, non-enveloped. So basically the naked capsid, in theory, is open to the immune system for attack by antibodies. And Richard did a great job of talking about the evolutionary sequence space there. But what we've known for years is that when you infect a cell with Picornaviruses, you get these unusual double membrane vesicular structures. We've known about them for years, and on the right, there's an electron micrograph that was taken by George Palati's lab in 1965. This is of a poliovirus-infected cell, and you can see those really unique double membrane structures. And when we see these structures, we find that it looks like there's almost cytosol inside these double membranes. So you see 2 lipid bilayers with a little space between them, and then almost what looks like cytosol. The other feature is we see virus inside and near these structures, and no one really understood why. And Palati actually hypothesized that this was part of the innate immune response, that this was getting rid of viruses. We have observed these double membrane vesicles during infection with EVD68. So when we look at uninfected cells on the right, you don't see any structures like this, and they start to form at 5 or 6 hours after infection with EVD68. And as you go from right to left, you see increasing magnification. And on the left, you can really see those double membrane structures with 2 lipid bilayers and then inside sort of an electron light contents. Now, it's not a new concept that maybe vesicles could be involved in Picornavirus, and especially in Picornavirus exit. These images taken by Robley Williams' lab in 1969 really strongly suggested that there was vesicles fusing with the plasma membrane to release virions. And you can see, if you look at the upper right, it really looks like the result of an event where a vesicle fused with the plasma membrane and released 3 virions. But the problem is that we were seeing double membrane vesicles, and that topology doesn't make sense. If a double membrane vesicle fuses with the plasma membrane, you'd expect to have a single membrane structure released from that cell that would contain multiple virions in it, and we just didn't have any evidence for this happening. Until a few years ago, when for hepatitis A virus, another Picornavirus, Stan Lemon's lab discovered that there's an enveloped form of this virus. And he has shown that this envelope is derived from the endosome pathway. It actually requires the escort machinery, which is part of endosomal trafficking. And he also showed, well, he hypothesizes strongly that this is primarily a form of the virus that exists in a host when the virus is trying to traffic from cell to cell within a host and avoid the immune system. and that the naked form that we've been typically studying is really the form that it goes from host to host. And it's a lot hardier and a lot more, again, open to the immune system. And a few years later, Ralph Foyer's lab, along with collaborators, showed that for Coxsackie virus, yet another picornavirus, these envelope forms were derived from the autophagy pathway. Now, I'm going to explain autophagy to you in a minute, but I can tell you that this got us very excited because double membrane vesicles are the hallmark of the autophagy pathway. So we thought maybe our pathway has something to do with this envelope form of the viruses we were seeing. So what is autophagy? Those of you who haven't heard of autophagy before, this is a process of literally self-eating, autophagy. Cells are digesting themselves, and it's an important part of how cells maintain their homeostasis. This is how organelle turnover occurs. So it's critical during development. It's critical during organismal development. It's typical in cellular homeostasis. It's important for a stress response, especially when a cell is starving. It will use autophagy to respond and generate new amino acids. But it's been implicated in a number of diseases, and limited autophagy is a factor in neurodegenerative diseases where aggregates aren't cleared from neurons, digestive diseases such as Crohn's. It's been implicated in multiple cancers, and it's also been shown to be an anti and pro-microbial event. So how does the autophagy pathway work? Well, what happens is you get these crescents that form and encompass cytosol. And those crescents then are called autophagosomes, and they fuse with endosomes. And the endosome delivers its contents, and now the autophagosome becomes acidic. We call that an amphosome. The amphosome is then capable of fusing with the lysosome, forming an autolysosome, and that's where the cellular contents that were entered to the beginning of the pathway are actually degraded. There are two proteins I'll just mention on this slide. One is the LC3 protein. It's critical for autophagosome formation, and it becomes lipidated, and that lipidated form is then found on the pre-autophagosome crescent. And then there's a second protein called P62. This is a cargo adapter. It has another name, SQSTM1, and it is actually binds to cargo and is degraded with the cargo. So we use these to monitor the autophagy pathway, and they'll come up in a couple of future slides. So our first question was really, does this represent an antiviral response that degrades variants? Pretty much the question that George Plotty was asking in the 60s. And we found that when we starve cells to promote autophagy, we also promote replication of EBD68. So if you'll see here, we're starving the cells, and we get about 2 to 3-fold more virus, both within a cell and extracellularly. And we can verify using that P62 SQSTM1 protein that we are indeed inducing autophagy with starvation. I'm not going to walk you through all of the data, but we've shown this in multiple ways for both EBD68 and numerous other picornaviruses that the cells actually thrive from the induction of autophagy, which we found a little surprising at first. So we wanted to know, well, wait, it's inducing this pathway, but is it actually activating autophagic degradation? I mean, is the virus actually turning on this degradative pathway? And if so, maybe there's an antiviral factor being degraded. And the second question, of course, would be, we see virus inside of these vesicles. So if the virus is inside these vesicles and it's being delivered to lysosomes, how does the virus survive the autolysosome? So we attack these problems by looking at some SNARE proteins, and there's a known SNARE complex that's involved in the fusion of the autophagosome with the lysosome. And the key member of that complex is called SNAP29. SNAP29 bridges the two vesicles. So when we looked at SNAP29, we actually are looking at a regulator of autophagosome membrane, lysosome membrane fusion. And when we look during infection, we actually could find a cleavage product. If we look down here and down here, during infection, we could see that this SNARE protein is cleaved. And that was interesting. We can actually see it with two different antibodies to the C and N terminus. So if you look in the bottom right here, you'll see that we could map potential cleavage sites. And when we overexpress the 3C protease from the virus, we can recapitulate that cleavage without the virus. So the virus seems to actually be attacking this key protein that allows for fusion of autophagosomes and autolysosomes, which means that we don't think this is progressing. We think the virus is actually preventing the autophagosome from getting to the lysosome. This is a really great idea for potential therapeutics, because if we could maintain SNAP29, theoretically, we could actually target this virus to the lysosome and we could degrade it, and then we would get a reduced infection. Now, we had another question, which is, well, why? Why the autophagy pathway? Why enter a pathway that is a lysosomal fate if that's not what the virus really wants to do? And so we knew it wasn't the lysosome. So we decided to ask, well, is it this acidification step that's upstream? Is the virus somehow benefiting from acidification of the vesicle? And we have a couple of ways we can block acidification. One is a drug called baflomycin A1, and another is just treating the cell culture with ammonium chloride. And when we blocked the acidification with ammonium chloride and we did an EVD68 infection, we found that we lost about a log of infectious virus. The extracellular, interestingly, was not particularly affected. It might have been slightly bumped. It's not very significant. But the cell-associated virus was dropped by about a log. So we're losing something like 90% of infectivity. And we could show and see that this actually was having an effect. So we wanted to know which step in virus production was being promoted by vesicle acidification. Very simple life cycle. These viruses get in, they translate their genome, they cleave their single protein into lots of other proteins, they make RNA, they make capsids, the RNA goes in the capsids, the virus exits. So we wanted to know, well, where is the virus using this acidification? Where is it using vesicles? So we started by doing a simple question of, we know that this virus turns off translation very efficiently. So if we do a pulse chase, cellular translation is gone and only viral translation remains. So we're using S35 methionine to pulse chase here, and we can actually show only the viral proteins being produced by about 1 to 2 hours after infection. And we can see that they look like the typical viral proteins we would see in an infection historically. And when we inhibited acidification, we found that we saw no change, that we saw those same proteins at that same rate, which meant the virus was getting in, translating, and processing its polyprotein at the usual efficiency. So then we looked at RNA, and we asked, is the viral RNA affected by vesicle acidification? We blocked acidification, and we got about the same amount of viral RNA. We can block acidification with we can block RNA replication with other drugs. So we knew that acidification was not having any effect. And we did show that we are in fact still getting that 1 log drop in virus, even though we're not getting any change in protein or RNA. So we were a little puzzled by this. And so we decided to look to see if we were getting the same number of capsids. And we can get both empty and genome containing capsids. And we can separate them because the RNA changes their sedimentation velocity, and we can find the empty capsids and we can find the capsids that have RNA. And when we did this experiment, I'll just tell you, we did not see a major effect on any change of whether there were capsids that were empty or capsids that have RNA. So far we know that we're losing 90% of infectivity, but we're getting the same amount of RNA, virus, and capsids with RNA in them. So what was left? And there was one step left. And that was up here in red, there's a cleavage maturation that occurs where one of the capsid proteins is actually cleaved, and that makes the virus infectious. And so we decide we can't actually separate those two because they have the same sedimentation velocity. So we took that fraction and then we looked at the capsid proteins to see if they were changing. And what we found, if you look over here, it's clearest to see in the 6 hour. You can see that this VP0 protein, which is completely gone in a normal infection, and VP2, which is highly prevalent in normal infection, when we inhibit acidification, now we can see a VP0 band, and VP2 is strongly reduced. So this told us that the only difference we were really seeing in these viruses where we were losing 90% infectivity is we were losing the maturation, and that's it. And that's really interesting because that means that we're generating capsids that could theoretically be antigenic, but they're not infectious and they're not dangerous. So we're dropping 90% of infectivity, but not 90% of the antigenic capsids. And we're really excited about that also as a potential treatment and maybe a potential safety mechanism for a vaccine. Okay, so with that, I'll tell you a very brief wordless summary. This virus gets in, it generates these autophagosomes. Some of the virus winds up in these autophagosomes. Those autophagosomes acidify, and the virus now becomes more infectious. It promotes the infectivity. Normally, these would go to the lysosome, but they do not go to the lysosome in this case. So the virus blocks that, and we think that the virus exits using these vesicles, and that generates both free and membrane-bound forms of the virus. And that's my summary. Basically, I just have listed it here. You're welcome to read, but we're a little bit behind, so I'm just going to blow through the summary. And I'm just going to say we think this is an interesting possibility for both therapeutic and vaccine targets. These are the people who did the work. I especially want to point out Alexia Richards, who did a lot of this work. And I want to mention that we have a postdoc available and we're completely funded. So if you can move to Maryland and wear a mask, we want to have you in our lab. And with that, I am happy and excited to introduce my colleague and friend, Amy Rosenfeld. Amy is an assistant professor in the Department of Microbiology and Immunology at Columbia. And Amy is going to tell us a very exciting story about enterovirus D68 and the development or not of neurotropism in this virus. So with that, I turn it over to Amy.